Good afternoon, Chris or Pri. Today I'd like to delve into our Week 8 practical, our fourth experiment, and I'm going to focus on the first and third sections, or A and C, in relation to proteins and amino acids, and specifically a protein in milk called casein. In this section of our practical experiment, we use the Bradford protein assay, or Bradford reagent, to help formulate our results. Just to give you some background, the Bradford reagent was developed by Marion M. Bradford in 1976. It's a fast and accurate way to measure the presence or concentration of certain proteins in a solution. It does this by reacting to a specific R group, which is attached to the amino acids that the proteins are made of. There are 20 different amino acids that are found in proteins. They all have different R groups, so with the correct reagent, in this case Bradford's, we can identify, as I said earlier, the presence and strength of certain proteins and the amino acids in their makeup. In practice, for our experiment, we just looked at milk, preferably skim milk to avoid the fat content separating alongside the casein. Just ordinary skim milk, I would assume it was pasteurized as well and bought from a store, as opposed to straight from the cow, of course. Now, in milk, the dominant protein is casein, and that's what we're looking for. Casein, in its proper form, is 224 amino acids, and while that doesn't really work with the 20 amino acids I mentioned earlier, of course there are some repeats. Now, back to the Bradford's reagent. Bradford's reagent contains a dye called Koumassi Brilliant Blue G250, which reacts only in the presence of some of the specific R groups that I mentioned earlier. In terms of casein, only 13 of the 224 found within casein react with Koumassi dye. Kindly given us to the lecturers in the prac book, those amino acids were 1 tryptophan, 4 arginines, 4 tyrosines, and 4 histidines. And upon contact with any of those amino acids, the dye reacts following its namesake and turns a clear blue color. Actually, depending on the ionic state of the dye, it can be blue, green, or red in anionic, neutral, or cationic situations respectively. But for the purposes of this experiment, it turns blue in the presence of any of the mentioned amino acids. The greater the concentration, the deeper the blue. So now the background information is out of the way, how is all of this used in practice? First, to isolate the casein from the milk, the components had to be separated, which is one of the reasons why skim milk was used, because fat is just one of the components removed during this section. Because there was no fat, the only thing that had to be separated was the calcium and the milk sugar lactose. The simplest method to do this was to add an acid to the milk to remove the calcium, and as a side benefit, when this is done in a warm solution, the casein separates and somewhat solidifies in a large blob or colloid in the solution. After this can be removed physically with some care, leaving the lactose behind in a yellowy sort of solution. The acid here was used was an acetic acid and had to be added in careful increments, otherwise parts of the casein could redissolve back into the solution. Now, after this was all completed, we dried and filtered and sent it through some water and then filtered it again, and then weighed to produce a value for the yield. And in this case, with 200 ml of skim milk, we should have received a yield of 4.6 grams, but instead received about 6.787 grams. The casein perhaps could have been dried for longer between the filtrations to make sure there was only casein and no water present at all, but in this case it was not maybe done adequately. And that was our process for isolating a specific protein from a solution, in this case milk and casein, and while not perfect, it helped us to gain an understanding of how proteins in solutions function and what can be achieved. Now, Part B focused on separating and identifying different amino acids in a solution using a process called thin layer chromatography. However, to keep the focus on Bradford's reagent and casein, I'll skip to Part C, where we determine protein content in different samples actually using the Bradford's reagent. During Part C, half of the section was to examine concentration levels in milk and increasingly dilute concentrations to fit to a curve. And the second section was to compare and determine the protein content of milk against a high protein milk drink such as Up and Go. And for the first half, 1 ml of Bradford's reagent was added to 20 microliters of 7 different prepared concentrations, being 2 milligrams per liter, 1.5 milligrams per milliliter, 1.0 milligrams per milliliter, 0.75 milligrams per milliliter, 0.5 milligrams per milliliter, 0.25 milligrams per milliliter, and finally 0.125 milligrams per milliliter. After some simple mixing of these concentrations, they were then measured by an attachment to the LabQuest 2, or Vernia, called the SpectroVis, which rated the differences in color, or the depth of the blue, I suppose, of the Bradford's reagent far more accurately than our eyes could determine, and then plotted it on a curve. And now, I don't have a screen behind me, I don't have the photo of the curve that was generated. Each decreasing concentration roughly fit the correct values in relation to one another, and the table can be found on the practical video for that week. For the second section, comparing the protein concentration between the milk and the high protein Up and Go drink, it was far simpler. A measurement of 980 microliters of phosphate buffered saline, or PBS, to 2 different tubes, and then 20 microliters of milk and high protein drink respectively. After this, 2 cuvettes were prepared with 1 milliliter of Bradford's reagent each, 20 microliters of the high protein PBS solution and milk PBS solution were then added to the respective cuvettes, mixed and measured, also using the LabQuest 2 SpectroVis. In our results, the high protein drink contained more than double the protein content of the regular milk of the same volume. And it just shows that we learned so much. Anyway, these experiments were important to us in our learning as they showed us firstly the style of experiments we may be called upon to do, the style of experiments that are effective for receiving and comparing different results, as well as teaching some of the more delicate physical nature to performing scientific experiments such as these. Thank you.